and the leader of the field of population genetics in India. Professor Partha Pratim Majumdar, National Science Chair, Government of India. And we are so fortunate that today he is our guest speaker, who dedicated his career to developing methodologies for mapping human disease genes, identifying factors underlying these many human genetic diseases, notably for oral cancer, and reconstructed the ancestries and the relationships of various endogenous population groups of India and Asia with the help of genomic tools and its science. He has immensely contributed in the capacity building of human genetics and population genetics in India. Sir, I myself, and on behalf of the RGU, extend our gratitude for graciously accepting our invitation and visit to us and also share with your expertise with us. Professor Majumdar, lovingly I call Partha, who obtained his BSc, Master's degree and doctoral degree from Indian Institute of Science, Kolkata. And after that, he left for his postdoctoral research at the Center for Demographic and Population Genetics, University of Texas Medical Center in Houston. After this, he served as a faculty in the Department of Biostatistics and Human Genetics of University of Pittsburgh and US. His major scientific interests and contributions have been in the field of human genetic disorders and population genetics. He is the founder of the National Institute of Biomedical Science at Kalyani, and then he was a director of that. Currently, he is an emeritus professor of Indian Statistical Institute, Kolkata. He is a member of the organizing committee of the International Human Cell Atlas Consortium and in the Indian National Coordinator of the International Cancer Genome Consortium. He has provided service to UNESCO and WHO. He is a recipient of many awards and medals, just to name a few: the TUAS Biology Prize, Golden Jubilee Commemoration Medal of Indian National Science Academy, G.D. Birla Award for Scientific Research, the Ranbaxy Research Award in Applied Medical Science, and the New Millennium Science Medal, Government of India. He has been awarded the G.N. Ramachandran Gold Medal by the CSIR. Friends and my colleagues, it's not just his professional achievements that make him very special. He is a passionate advocate for educating the next generation on the importance of human genetic disorders. His engaging speaking style and ability to connect with the audience of any backgrounds, make him an inspiration to many. Today, he is going to deliver his talk on Genomics of Cancer, a big bouquet of stories. And I'm sure that this will be an intellectually stimulating and offers a unique opportunity to the students, faculties, and the members of the community to engage with cutting-edge research and ideas. Professor Partha, we are requesting you for this.
Request our Honorable Vice Chancellor, Professor Dr. S.P. Singh sir, to please felicitate our guest of honor.
This talk. I'm very, very thankful to the Vice Chancellor, to Professor Anupam Chatterjee, and to Professor Mihir Chaudhary. I've known them for many, many, many years now. I will start with an apology. This talk is meant only for the people sitting at the back. It's not really meant for the people who are sitting in the front. It's not meant for faculty members. It's really meant for the students in this hall. And what I'm going to do is essentially to tell you a set of stories. Hopefully they will inspire you, they will attract you to the field of genomics and to do more research, to figure out more nuances of cancer as a disease. And that's what I've set myself to, that's a task that I've set myself to. And I hope that at the end of the talk, you will be convinced that these stories are interesting and some of you may actually be inspired, like I said. I have removed 2 words from my title. The title that I had given to Professor Chatterjee was Genomics of Cancer, a Bouquet of Stories. And the reason why I've removed two words from the title is I didn't actually want to scare the students away. So it's really, just relax. It's really a bouquet of stories, very, very inspiring from the people who have done the kind of work that I'm going to narrate to you, and hopefully we'll all be able to contribute a little bit towards understanding of cancer. I will invoke your imagination a little bit and just imagine if you could charm a bullet to actually find out and kill a person. This is charming a bullet, right? And such a bullet is usually called a magic bullet. I don't know if you can charm a bullet, a real bullet, to go and kill a person, and that's not the intent. We don't really want to kill a person, but we could call such a bullet as a magic bullet. And an old superstition believed that such a bullet could be made, such a magic bullet could be made, and it was indeed possible to have such a bullet which would be called as a magic bullet. There is a reason why I'm telling you this story. It's really a superstition, but in the field of science, in the field of medicine, this has really become possible. In the 1900s, medical scientists discovered that microorganisms such as staphylococci and streptococci cause disease. So this was not even known at that time that microorganisms could cause disease. Today we just take it for granted, right? Many of these infectious organisms cause disease and we take that for granted, but as a matter of fact, in the 1800s this was not even known that it was a major discovery. Microscopes were invented and they began to investigate them with microscope. And here is a person whom we should remember. He eventually got a Nobel Award, Nobel Prize. He lived from 1854, passed away in 1915. His name is Paul Ehrlich. He actually, discovered the fact that these microorganisms could actually cause disease. He searched for chemicals. In addition, he searched for chemicals that would stain specific germs to make them more visible under the microscope. That's what we do most of the time now. We stain specific microorganisms with specific chemicals and then find a chemical that would both attach itself to the germ, that is stain the organism, and also kill it. So this is what we try to find, that we not only are able to identify the organism, but because it causes harm to us, we actually want to kill it. And what he said is that such a bullet, this is called a magic bullet, and essentially you not only are able to target the organism, but also simultaneously kill it. So you find your target and also kill the microorganism. So Ehrlich introduced the term magic bullet in this kind of literature that has to do with diseases. We'll come back to magic bullets later. However, on the one hand, one is looking for a specific target, one is looking for targeted killing with magic bullets, but on the contrary, there are other people who think otherwise. And I'm going to show you the picture of a pretty lady whose name won't be written, but most of you will be able to recognize, at least most of the people at the back. Maybe some people in the front will not be able to recognize her. And this is the lady. Is there anybody who doesn't recognize this lady? No hands raised, right? Everybody recognizes this lady. Why am I talking about this lady? Not because she's pretty, not because I'm in love with her, nothing of that kind. There is a specific reason why I'm telling you the story of this lady. This lady, her name is of course Angelina Jolie, all of us know. Her mother's name was Marcellina. She died at a relatively young age of 56 from ovarian cancer. For ovarian cancer, for people to die, women to die of ovarian cancer, 56 is a relatively early age. And many of us know that, when you have cancers early on, you probably have inherited a gene from somebody, one of your ancestors, and you're likely to pass it on to your daughters and so on and so forth. So what she did, what Angelina did, she knew about this fact that she could have inherited the faulty gene from her mother. She got herself tested and indeed figured out that she indeed got a faulty gene that causes ovarian cancer and other cancers as well from her mother, Marcellina. So one of the first things that she did, she said that, this particular gene, her doctors told her, not only predisposes to ovarian cancer, which is how her mother passed away, but also predisposes a woman to breast cancer. So she said that, what I'm going to do, I'm going to get rid of the organs from my body that will actually get affected. So she underwent what's called a radical mastectomy, removed both breasts from her body, such that if the organ didn't exist, then the disease would not come. This is what she did. She got rid of her breasts, and the gene that she actually inherited from her mother is called BRCA1. BRCA, this particular gene, is on chromosome 17, is near the centromere on chromosome 17, and it not only increases the susceptibility to ovarian cancer, but also to breast cancer. There is yet another gene called BRCA2, which is on a different chromosome, chromosome number 13, that also increases the risk of both breast cancer and ovarian cancer. So Angelina Jolie actually inherited a variant in BRCA1, this particular gene that enhanced the risk of breast cancer. She got rid of her breasts. So that's a fault in a gene, and that's essentially what we shall be talking about. Let's look at the kind of risk that one entails if one inherits a gene variant in a gene called BRCA1. In the general population, the lifetime risk of breast cancer is about 12%. If you have inherited a BRCA1 variant, if there is a variant that you have inherited from your mother, the risk increases by about 5 times, and this is the lifetime risk, and this 5 times increases only by the age of 70 years. So its lifetime risk is even more. For BRCA2, the risk is about 4 times. This is not only true of breast cancer, but it also enhances the risk of ovarian cancer by about 4-fold, for 3 to 4-fold for ovarian cancer, and about 1.5 times for BRCA1 variant for ovarian cancer. So since Angelina Jolie knew that this is what she had inherited, she got rid of her breast. She underwent a radical mastectomy and got rid of her breasts. And I will come back to this statement. And not only that, a couple of years later, she got rid of her ovaries and fallopian tubes. So essentially what she was telling herself, I want to reduce the risk to these kinds of cancers by removing organs from my body. So on the one hand, we have the Nobel laureate Paul Ehrlich, who is actually talking about targeted therapy. On the other hand, this lady is talking about doing things to her body such that she's just getting rid of the organs. When I read these kinds of stories about Angelina Jolie, I wondered for myself, there are now genes that predispose to lung cancer. What happens if she knew that she has inherited a gene for lung cancer, from one of her parents? What would she do? She couldn't possibly get rid of the lungs. Then she wouldn't even, live. And so there is a limit to how much you can use this kinds of information for carving your own body. Essentially, she's carving her own body, and that's not the right approach, all of us will agree. I mentioned, you may or may not have read that, I mentioned at the bottom of one of the slides that BRCA1 is a tumor suppressor gene. Tumors, there are two classes of genes that cause cancer. One class of genes is called tumor suppressors. These tumor suppressors are up and awake all the time. And they actually produce proteins that are very useful. They are useful because they keep tumors in check. They don't allow tumors to happen. These are normal growth inhibiting proteins. They keep cell division under control, and I'll talk a little bit more about cell division in a minute. So these tumor suppressor genes are awake all the time, and they are doing good things to our body. One good thing that they do is that they don't allow tumors to appear. And sometimes there is a mutation. A mutation is an alteration in the DNA underlying that particular gene. If there is a mutation, then the protein is not produced. The normal protein is not produced by that particular gene because of the variant, because of the alteration. Then it doesn't do its proper function, or it goes to sleep. It doesn't produce any protein at all. So it doesn't produce any good protein, and it doesn't, or may not even, or may produce a bad protein, or may not produce any protein at all. So if there is a variation, then it may not produce any protein, or it may produce a faulty protein. In any case, what happens is that it's unable to carry on its normal function of keeping tumors in check. So these are called tumor suppressors, suppressors, and variations here will lead to development of tumors. So a mutated tumor will produce a defective, non-functional protein. So let me say a little bit about cell division. So cell division, as most of us know, is 1 cell to 2 cells. That's a normal cell division. 1 to 2 to 4 and so on. 2 to the power of n division is what? So it's like a binary fission. Hallmark of cancer is that this particular kind of cell division goes completely haywire. So essentially, one cell, instead of producing 2 cells, might produce 10 cells. And one of those 10 cells, again, might produce another 20 cells, and so on. So this is how one hallmark of cancer is unregulated cellular growth. Normal cell division is regulated cellular growth. Hallmark of cancer is completely unregulated cellular growth. And the kind of proteins that are required for regulated cellular growth are produced by these genes that are called tumor suppressors. And when these genes become defective, they don't produce the kind of protein that's required, and therefore the normal regulation of cell division goes haywire, and then it becomes, then the cells start going through an abnormal phase of growth. There is another class of genes that are called oncogenes, and this is the second class of genes that we know that produce that are related to cancers. And the oncogenes are like sleeping giants; they're sleeping all the time. They're not producing any protein. But somehow, if you activate them by some kind of a variation, some kind of a mutation in the gene, they will start producing a protein or producing different kinds of proteins. These oncogenes, and those proteins are rogue proteins in some way. They will actually cause cancer. So on the one hand, you have a class of genes that are always active, but you make them go to sleep, then you get cancer. Another class of proteins, they are always asleep. If you wake them up, you get cancer. So these are the two classes of genes: tumor suppressors and oncogenes. So we'll remember this. I'll come back to these tumor suppressors and oncogenes as I go along. However, like I said, that variation in genes, variation in the genome, variation in these classes of genes are responsible for causing cancer. However, if you look at cancers, only about 10% of any cancer is due to inheritance of a faulty gene. So if a mutation takes place, that particular mutation is passed on to the offspring and causes cancer in the offspring only about 10% of the time. So what it means is that when you talk about a genetic disease, and if I make the claim that cancer is a genetic disease, one would tend to think that cancer will aggregate in families, cancer will be passed on in a systematic Mendelian fashion from parents to offspring and so on. It doesn't happen that way. It only happens in about 10% of cancer cases where there is this neat kind of transmission from parents to offspring. That's only about 10%. Yet, I'm going to make the claim that cancer is a completely genomic disease or completely genetic disease, and I'll justify why I say so. I'll start with, so that was Angelina Jolie was my first story. I'll start with my second story and a third story and revisit the second and third stories towards the end of my talk. The second story is, these are not made-up stories, by the way, as you could see. Angelina Jolie's is not a made-up story. This is also not a made-up story. This is a picture that appeared in the oldest medical journal in the world, called the New England Journal of Medicine. Most of you would not know what this picture is. This is called a PET scan. And essentially, it's an image of the internal organs of the body, and it actually takes images of the soft tissues within the body. You see lots of black dots. These black dots are all soft tissues, and they're all tumors. So this individual is completely filled with tumors all over the body, as you can see. Of course, there are one or two organs, as we'll see later, but wherever the tumor appeared, that has spread throughout the body and completely engulfed the entire body of this individual. This person came to a doctor, and the doctor said, well, I'm going to treat you. I'm going to provide you with a medication at a particular dose, and be on the medication for a certain length of time, 3 weeks, and then you come back to me and we'll do another image of your body and see what happened to the tumors. So this person, the doctor did that, and the person came back after 3 weeks after having taken that medication, and this is what the image looked like. All the tumors have evaporated. This is the kidneys, and so that's why there is a black dot. But all the tumors have evaporated from the body, and this is what we see of the individual. So this is a very happy tale that the treatment actually worked, and the treatment actually provided complete amelioration of cancer to this individual. 2 years later, another patient came to the same doctor with essentially the same kind of picture, and I'm not showing you a different picture because it was essentially the same kind of picture. And the doctor said, oh, I know how to treat you. I'm going to put you on, I've seen one such patient very recently, and I've completely treated that patient. So you come, I'll give you the treatment, you get yourself on this particular medication for 3 weeks, and come back to me and I'll show you what happened. All your tumors will be gone. That was the hope that the doctor gave to the patient. And this is what happened. Absolutely not even one tumor evaporated, not even one tumor. Every tumor that was there has remained in that particular position, and this treatment did nothing to the tumors in the body. So the question that we ask is, why is there a smiley and a sad face here? Why is there a smiley here and a sad face here? We'll revisit this story. We'll remember this story, we'll revisit. My third story is a personal encounter. Like you have heard that I did most of my studies at the Indian Statistical Institute. Went there after I passed my high school and spent my life there, essentially getting my degrees and then serving on the faculty of the Indian Statistical Institute. At the Indian Statistical Institute, you learn statistics is the major, but you also learn various other kinds of sciences, primarily because our founder believed that unless you learn the sciences, you cannot apply statistics. Statistics is basically an applied science. So we had to learn various kinds of sciences, including physical sciences, biological sciences, and so on. So this pertains to a teacher of mine, a teacher who taught us geology. We knew that he was a chain smoker, and this is the story. He retired. I got to know the family well. He retired, and this is the story that I'm mentioning to you. After he retired, he actually got lung cancer. And his wife contacted me because I had started doing work on cancers by then. So she said that your teacher has got cancer. Can you help me? Put me in touch with a cancer clinician in the city of Calcutta, and I want to get your teacher treated. So I said, yes, I will. With a cancer clinician. And one day, both she and I, we went to the clinician, and both of them were walking along the corridor, and I was sort of tiptoeing them from behind. I overheard a conversation which I want to share with you. The doctor says, well, the patient has, don't worry about what this is, non-small cell lung carcinoma. The patient has lung cancer. I need to put him on chemotherapy. As soon as the wife hears the word chemotherapy, her tentacles go up. because she knows what chemotherapy will entail. The husband will lose his hair, will not have taste, and will refuse to eat, and so on and so forth. So she has to be by the side of her husband, and she lives about 40 kilometers away. And in the city of Calcutta, kilometers are much longer than in the city of Guwahati. You take a long time to traverse a kilometer, primarily because of traffic jams and all that. So to traverse these 40 kilometers, she would need about 2.5, 3 hours every day coming into the hospital and going back. So she asked a natural question. She asked, can the chemotherapy be done at home? The doctor sort of gets angry. She said, no. But then doctor says, wait a minute, let's check out his EGFR mutation status. EGFR is a gene, epidermal growth factor receptor, and I'll tell you a little bit about what this gene does later. So he says that let's check out his mutation status, and goes on to say that if it turns out that your husband has EGFR mutations, then you can take him home and administer chemotherapy orally. So now the wife is really praying that her husband has mutations in the gene. Just a little while ago, we talked about mutations, and we said mutations are bad. Mutations in tumor suppressors are bad because, they go to sleep and the tumors will form. Mutations in oncogenes are bad because they will wake up and produce bad kind of proteins. And here you see what the doctor says. If he has mutations, then you can take him home and administer chemotherapy orally. We'll revisit this story in a few minutes. So we'll revisit story number 2 and story number 3. Cancers, if you didn't know, have a very disorganized, normally cells are very organized, 23 pairs of chromosomes, they occur in pairs and so on and so forth. So this is a normal set of chromosomes in a normal cell, and they have a very regulated cell growth. Of course, most cells contain a copy of the entire human genome and so on. Cancer cells, on the other hand, are very disorganized, as you can see from this picture. Here there are 2 yellows, and here there are multiple yellows, and not only full yellows, but yellows with reds and greens and so on and so forth. So as you can see that there are not 23 pairs of chromosomes, but many more. And this disorganization leads to unregulated cell growth, and the hallmark of cancer is unregulated cell growth. So I'm going to discuss a few types of cancer and tell you a little bit about these kinds of cancer. Worldwide, about 1.6 million new cases of breast cancer are diagnosed each year, leading to over 500,000 deaths. And we talked about Angelina Jolie and her mother and so on and so forth. 40 years ago, 50% of women who diagnosed with breast cancer would survive for more than 5 years. Only 50% of women would survive for more than 5 years. Today, that 50% has changed to 80%. So 80% of women who are diagnosed with breast cancer will actually survive for more than 5 years. Now, this is not magic. There is intervention, human intervention that was required. And so the question is, how did this change happen? In 40 years, an increase of survival by 30%. How did that happen? So let's just look at one subset of these kinds of cancers, these kinds of breast cancers. In about 20% of breast cancers, the cells make too much of a protein known as HER2. This is a protein that's, of course, coded for by a gene, and it produces that kind of protein. And once it produces this kind of protein, and this is due to the overproduction of this kind of protein, then those breast cancers seem to be very fast growing. Once this was understood, it was not, one needed to do a lot of scientific research to be able to figure out that, that these cells, in about 20% of breast cancer, cells make too much of a protein known as HER2. Once you identified that, then what does HER2 do? Why do these cancers tend to be fast growing? So where is HER2? HER2 is very close to the BRCA1 gene on chromosome number 17, and BRCA1 is one of those genes that actually, one of those tumor suppressors, which if mutated will lead to breast cancer. HER2 is another gene which occurs in multiple copies, but when the number of copies become large, then they start producing a lot of protein, and that protein becomes harmful to the woman, and it leads to breast cancer. This is called a proto-oncogene or an oncogene, as I told you, and I've already told you this, that these are sleeping giants. Once they start overproducing these proteins, then there is abnormal cellular control, and therefore they will accelerate cell growth and division, which is really a hallmark of cancer. I'm not going to go through all of the details. This is the structure of HER2 or HER2 protein, and because this overproduction led to breast cancer, we needed to stop that overproduction, or at least not have all of the protein function. And so what a molecule was identified that would attach to this particular protein called HER2 and not allow the huge amount of protein to actually function, because if it attaches to HER2, it will stop the protein from functioning. So that's really like a magic bullet, because now this particular molecule that we are talking about is actually attaching itself specifically to a particular protein called HER2, and then because it is attaching itself, it's not letting letting the protein do its normal function, as a result of which, because it's an oncogene, if it does the function, it creates the tumors, and so by suppressing it, you're actually controlling cancers. So it dramatically reduces the risk of cancer coming back and has very fewer immediate side effects. Chemotherapy, all of us know, you lose hair and then you lose taste buds and so on and so forth. So it also has dramatically far fewer side effects. So this is really like a magic bullet. Like I mentioned in the beginning, it's targeted, it's attacking a particular protein and saving human lives. So essentially, it's saving lives of women by tailoring drugs more precisely, and the information comes from research in genomics to be able to identify that the HER2 gene, the number of copies of HER2 gene is increasing, and that's the reason why there is overproduction of that gene. Then you do a lot of structural biology to find the structure of the protein. Having understood the structure of the protein, you know which particular place in the protein that you need to attack, such that it will become non-functional. All of this was hard work by years of hard work by a large number of scientists to be able to get to this magic bullet called Herceptin. So it acts like a magic bullet, but honestly there is nothing magical about it. It's hard scientific work to be able to understand the disease, to be able to understand the cause of the disease, to be able to figure out how to control that, and then eventually identify the molecule. There's nothing magical about it. Now my next story is going to be about children. I'm going to talk about children, and this is very, very dramatic, extremely dramatic. This is the percent of surviving children with one particular form of leukemia. Don't even worry about what that form of leukemia is. Leukemia is blood cancer, as we call it in the common day parlance. So it's percent of surviving children, which is on the y-axis, and this is years. So what you see is that in 1960, the percent of surviving children was 4. That means if there were 100 children with leukemia, only 4 of them survived. This is 1960. 2014, that figure has increased to 92%. 96 of these children would die, and here 92 of the children will survive, and this is in a period of about 50 years. Today, that figure, I don't have that figure, but that figure has increased to about 99.2%. So almost nobody dies. If a child has this form of leukemia, almost nobody dies. And how did that happen? This is not magic. How did that happen? Hard work, years of hard work. This is essentially, the reason why I'm showing you this is that this is what's called a fluorescent in situ hybridization. Essentially what it does is it paints the various chromosomes, and it allows you to find out whether the chromosomes are intact. Here the chromosomes are not intact. That's quite obvious because you see a green and a red and a yellow together. The green is one kind of chromosome, the yellow is another kind of chromosome, the red is a third kind of chromosome. They all have come together. Bits and pieces of different chromosomes have come together. So this is what we've seen, and in leukemia, there are these bits and pieces of different kinds of chromosomes. They are called translocation. A part of one chromosome gets translocated to another chromosome, and that phenomenon is known as translocation. This kind of translocation, and this form of leukemia is due to a translocation. This kind of translocation, or the translocated chromosome, is called a Philadelphia chromosome. And the reason why it's called a Philadelphia chromosome is because this was discovered in the Pennsylvania School of Medicine, which is located in Philadelphia, by someone, and I'll tell you about that someone a little bit more. And this someone actually discovered this translocation, and it's now called the Philadelphia chromosome. As you can see, that a complete book has been written on the Philadelphia chromosome. This, like I said, is a translocation. This is the normal chromosome 9, and this is the normal chromosome 22. What happens is that a portion of normal chromosome 9 gets translocated to normal chromosome 22, and this is a gene called ABL, and this is a gene called BCR. Because of this translocation, you get the BCR and the ABL genes come together. The BCR and ABL genes independently were producing proteins. Once they come together, they produce what's called a fusion protein. And this fusion protein becomes extremely dangerous and causes leukemia. This is, again, I'm not going to get into this. Having understood first of all, having discovered this translocation, that this is the cause for this kind of leukemia, having understood that the structure of the fusion protein, it was not a cakewalk, but again some years of work to be able to find a molecule that will not allow the fusion protein from functioning, that will actually kill the fusion protein as soon as it is formed. And this is what's called imatinib. That's the drug that was formed that will kill that fusion protein from working. And if you kill the fusion protein is the cause of this kind of cancer, the leukemia. And if you kill that protein from functioning, the leukemia will not form. So essentially, again, nothing magical about it. It's saving lives of children, using a magic bullet, but nothing magical about it. Hard scientific work, identifying a disease, trying to understand what the cause of the disease is, and then having understood the cause, how do you root it out is what scientists do and what scientists before us have done. The CML, chronic myelogenous leukemia, this is one form of leukemia that we are talking about. The world celebrates September 22nd as the chronic CML day. And the reason why it is celebrated is that on this day, this translocation was actually identified. And who has identified this? Her name is Janet Rowley. She passed away she was born in 1925, passed away at a grand old age in 2013. And because of this discovery, she's saved millions of lives of children. Because of that, the highest US civilian honor was conferred on her. It's called the Presidential Medal of Freedom, and this is Barack Obama in 2009 conferring on her the Presidential Medal of Freedom. So scientists do work very, very hard, and sometimes they get awarded by the president of the largest, one of the most well-known countries in the world. So again, it's fault in a gene. In cancers, most of these faults happen during the life course of a patient. Now, I do want to point out, and I had mentioned this earlier, that only about 10% of cancers are familial, meaning that you find Mendelian transmission of a gene from parents to offspring and through generations. 90% of cancers, you don't find that. The reason why you don't find that is because those cancers are not happening in the germ cells, the cells that participate in reproduction. Unless those cells, the mutations or the changes take place in germ cells, they will not be passed on from parents to offspring. These changes are taking place in the body cells of the individual during the life course of this individual. These individuals are born with good cells, complete. We have several trillion cells in our body. All of them look exactly the same. They are good cells. But as we expose ourselves to, let's say, the rays of the sun, we have no control, right? ultraviolet radiation seeps in, and we get DNA alterations because UV light causes mutations. When we grow up, many of us start smoking. Tobacco is a potential carcinogen. They will make DNA alterations, and eventually we get, those alterations will cause cancer. These are happening in body cells, and therefore they are not passed on to our children. So even though, with respect to these DNA alterations, with respect to cancers, these DNA alterations are happening in the DNA, or alterations are happening in the DNA, only about 10% of the alterations are passed on to the children. The remaining 90% are not passed on yet. All of them are alterations in the DNA, or the cancers are happening because of alterations in the DNA, and therefore cancer is a completely genetic disease. Contrary to what we have learned in school, right? We are learning that somatic mutations, these are mutations in body cells called somatic mutations. Contrary to what we learned in school, a genetic disease need not necessarily be transmitted from parents to offspring. So this is a broader definition of a genetic disease. So if you look at the evolution of a cancer genome, essentially what it means is that if you take a tumor and look at the cells of the tumor, what is it that you find? How did the cells evolve over a period of time is what evolution of a cancer genome is. So like I said, we are born, and imagine, if you will, that this green surface is like the surface of the skin, epithelial layer of the skin. Below the epithelial layer, we have a large number of cells neatly stacked, and this is exactly how they appear when we are born. Every cell looks exactly the same. As we pass through our life, life course, mutations or DNA alterations start happening. We're exposing ourselves through rays of the sun to other kinds of chemicals, to tobacco smoke, and so on and so forth. So these kinds of alterations start happening. Most of these alterations don't, we are completely oblivious to these alterations because they are not harmful at all. They happen. Yeah, that particular cell might die after a certain period of time, and so on and so forth, but we don't even get to know about them. Sometimes we do get to know about them because they cause unregulated cell growth, and unregulated cell growth is the cause of tumors, as I've repeatedly said. So this particular cell, for example, these 3 cells have gotten mutations. Again, on the top of mutations, or new cells also get mutations, and on the top of pre-existing mutations, you have mutations. Sometimes these mutations can cause unregulated cell growth, such as here. Here, this particular cell, which got a second mutation, after having gotten the second mutation, it didn't divide as 1 to 2 to 4. It made a large photocopy of itself. It made 4 copies of itself, and that's what you have. So that's a small tumor. These tumors are happening in us all the time as I'm talking. As I'm being exposed to this particular form of light, probably it's causing DNA alterations, and these DNA alterations are causing little tumors. We are not aware of them at all. This happens all the time in our bodies. And so here is another mutation that provided a little bit additional growth advantage to this cell, and it made 6 copies of itself. Again, too small for us to be bothered about, and we don't get to know this. So this process happens. So here is another mutation that took place. This red mutation that took place on the over this black and yellow mutation that already pre-existed in this particular cell. This mutation was different from all of the previous mutations because this red mutation gave this particular cell a huge growth advantage. It breached the epithelial layer and became a solid malignant tumor, as you can see. It gave that particular cell a huge growth advantage. It became a malignant tumor. While we may not be bothered about these, the whites and the blacks and the yellows that may have caused a little enhancement of the dysregulation of cellular growth, we need to be bothered about these red mutations because that's what has caused this malignant tumor. So to be able to understand these malignant tumors, we have to catch hold of these red mutations, and these red alterations are called driver alterations. What are these other alterations called, like the whites and the blacks and the yellows? They are called passenger alterations because they are not doing any harm to us. So what is driving the cell to becoming a malignant tumor is called a driver alteration or a driver mutation, and we are bothered about these driver mutations, but from the plethora of mutations that are happening in the cells, we need to identify these driver mutations, and this is not an easy chore at all. So it's like fishing out a needle from a haystack. not an easy chore at all. So there is a lot of statistics. First of all, we need to generate a lot of data on DNA alterations. Having identified some of these alterations, we now need to ask ourselves whether that's a driver alteration or not, and that involves a lot of statistical work. I'm not going to get into the technical details at all, but I do want to impress upon you that from the enormous mass of DNA alterations that we find, we need to identify these driver alterations. And as a matter of fact, even after a driver alteration happens, even in these cells that are a result of this driver alteration, there are more mutations happening, even in those cells. So the question that we ask is, what are the genes and genomic alterations that drive? Because we need to understand what are the genes and genomic alterations that drive, because unless we understand that, we can't even control it. We cannot find out a magic bullet that's targeted against that condition. So driving essentially means growth advantage, so we want to look at those DNA alterations that give a growth advantage. So that's the driver. I'm going to describe some work now. I've talked about women, I've talked about children, I'm now going to talk about men. And when I'm talking about men, I'm actually talking about some work that we have done. Our team was a large team. Teams, I must say, there was a clinical team that was that's located in Mumbai, in Advanced Center for Treatment, Research and Education in Cancer. Collaborator is Dr. Rajiv Sarin, who is actually the leader of the clinical team. And we had another team at the National Institute of Biomedical Genomics where we did all of this genomic work. The two people who actually led two arms of this study, I said that we need to generate a lot of DNA data. We need to be able to analyze the DNA data to be able to identify the driver mutations. So the person who actually was in charge of the lab is Dr. Arindam Maitra, and he generated all the data, oversaw the generation of all of this data. And after the data was generated, it was Nidhan Biswas who actually helped analyze all of the data and identified the driver mutations. I was the coordinator of this entire project, and I had my feet in multiple grounds. Let me describe to you what we did. This is what we did was oral cancer, essentially trying to find driver mutations of oral cancer. Oral cancer, it's the 8th most common cancer. It's about 250,000 cases appear, new cases appear annually, about 2/3 of them in developing countries. About 130,000 deaths happen annually, and it accounts for 1/3 of all tobacco-related cancers in India. Oral cancer is one of the drivers. Environmental drivers of oral cancer is tobacco chewing and smoking, so it's essentially tobacco-related. There is also a virus that is associated with oral cancer, but it's we have not found this association in India. It's essentially associated with tongue cancer that's been found in the West. That's the hazardous combination that's relevant to us. Tobacco, lime chews up the epithelial layer, and then the carcinogens in tobacco directly hit the cells and so on. So that's the hazardous combination. We looked at 500 patients. From each patient, we had sampled DNA or cells from the tumor and isolated DNA from those cells. We also had taken a blood sample and isolated DNA from the blood. And the reason why we did this paired way is because the tumor will have all of the mutations that were inherited, plus mutations that were acquired during the lifetime of that individual. The blood will have the inherited mutations but not the acquired mutation. So if we do both, if we sequence or generate data on both kinds, then we can subtract one from the other. We can subtract all of the mutations that we find from the blood from the tumor DNA, and then we'll be left with only those that are acquired during the lifetime of that individual. That's the kind of data that we analyze in order to identify driver mutations. Information of our patients, we are talking about a male cancer. 88% of our patients were male, 72% were middle-aged, between 40 and 55 years. Most of them presented very late when the tumor was very advanced. And the reason why most of these patients come to the doctor very late is because they love their tobacco. They will not give up their tobacco. So they find that there is a small tumor that has happened, small lesion within the oral cavity. They won't go to the doctor because they know that if they go to the doctor will tell them to stop doing tobacco. And they love their tobacco. They won't stop it. They don't want to stop it. So they come to the doctor when the pain is so large and the pain is so much, so great, and the tumor size is so large. That's when they come to the doctor, and that's essentially very late. Other than surgery, you can do nothing. And most of them were, of course, exposed to tobacco. We didn't find any evidence of bacteria. So I'm going to present data on 400. We now have data on 500. I didn't, update my slides, but again, this is what we do. We do whole genome sequencing, sequencing the entire repertoire of the genome that we have. But sometimes we also have done exome sequencing, which is really the genes that are there, the 15,000 odd genes that there are in the entire genome. The genome also contains a lot of extra material that's regulatory, that regulates these genes, etc. So when we just do the genes, that's called exome sequencing. When we do the entire genome, that's called whole genome sequencing. So this is what we have done. And I've mentioned that we have done both blood, DNA isolated from the blood, and DNA isolated from the tumor. And I've given you the logic as to why we do this paired sequencing. We have a very large number, a lot of variation across patients. I've talked about tumor suppressors. I've talked about what mutated tumor suppressors do. And the reason why I'm reminding you of this is that I'm going to just mention right now that what we have found are mutations in tumor suppressors. I've also mentioned about cancer. So what we did, well, after we analyzed our data on 400 patients, we found that there are 10 genes that drive, or alterations happen in 10 genes that are associated with the formation of these kinds of tumor, oral tumor. These genes have names, TP53 caspase 8, etc., TP53 caspase 8, etc. Some of these genes are more prominent than others because most of the cancer patients will have alterations in those genes. But again, I mean, it's 10 genes turn out to be significant drivers, and all of these are tumor suppressors. So essentially, all of these genes are active in oral cancer, and once there is a mutation or a DNA alteration, the protein will not be produced, and that's why they get these kinds of cancers. And if the protein is not, it's easier to stop the function of a rogue protein, of a mutated protein. It is very difficult to actually wake up somebody who was active and has gone to sleep. It's very difficult to wake up those kinds of proteins. So tumor suppressors is bad news. If a cancer is due to tumor suppressors, it's really bad news. I'll skip this. Let me revisit the two stories that I left unvisited. One is the story number 2, and the second is the story number 3. Story number 2 had to do with this, wonderful evaporation of tumors, etc. Story number 2 had to do with the conversation that I heard between my teacher's wife and the cancer clinician. I'm going to talk about a gene that's called BRAF. Mutations in this gene are found in 70% of malignant melanoma. About 30% of thyroid cancer patients will have mutations in this gene. 10% of colorectal cancer patients will also have mutations in this gene. So this is a gene that impacts on malignant melanoma. Melanoma is skin cancer, malignant melanoma in a major way, but it also is involved in other kinds of cancers. What this does, what this gene does, first of all, we need to realize that there is a lot of cargo that moves from outside of the cell to inside of the cell. And this cargo also controls cell growth. And let me see if I have a picture of this. Yes. So there's a lot of cargo that comes from inside of the cell to from outside of the cell to inside of the cell, and it's regulated by genes such as BRAF. There are other kinds of genes as well, but this is regulated, this kind of cargo, when it must come in and when this coming in of the cargo must stop. That's regulated by BRAF. And what does this cargo do? This cargo actually sends signals for the cell to grow. And when this, and it also tells the signal when to stop growth. Otherwise, it just will keep growing and it will become like a tumor. So this is what happens when there is a normal BRAF. There's regulated, import of cargo from inside to outside, signals the cell to grow, and that happens. When the growth is not necessary, the BRAF gene will signal, or it will stop, and it will signal the cell not to grow. When in a cancer cell, when the BRAF gene is mutated, the BRAF gene, when it's mutated, it does not signal the cell to stop sending cargo from outside to inside. Essentially what happens is that as a result of that dysfunction, the cell will continue to grow, and as I've repeatedly told you, unregulated cellular growth is the hallmark of cancer, and this is what happens. So there is the most prominent alteration that happens is an alteration at amino acid position number 600 in this particular protein, and that changes one amino acid to another amino acid. And now we know that there is because of this information, an inhibitor has been identified which is called vemurafenib, which actually is a drug that stops this impact of this mutated BRAF. And once the mutated BRAF is there, then it will stop the dysfunctional protein from acting because of this. And so we ask the question, why is there a smiley here and a sad face here? And the reason why there is a smiley here and a sad face here is because the doctor actually provided both of these patients with vemurafenib, which will act against mutated BRAF. This particular patient had a mutation in BRAF, and therefore this drug worked, because this drug is meant to work against that particular altered protein when there is such a mutation. This individual got cancer because of some other protein that went wrong, but the doctor assumed that this treatment would work, but the treatment didn't work because this individual did not have the mutation. So the mutation, the drug vemurafenib acts as a magic bullet only when there is this mutation in BRAF. When there is no mutation in BRAF, this is not going to work. The drug doesn't work. And that's exactly what happened. It worked so well in this particular patient, and it didn't actually work in this patient. So now we know when the magic bullet works and when the magic bullet may not work. So doctors now, even before they administer vemurafenib, these are expensive drugs. So you don't want to expose your patients if you know that the drug will not work. So the doctors now will actually find out whether there is a mutation for that particular patient, whether there is a mutation in BRAF, and only then they will prescribe this particular treatment called vemurafenib. Conversation that we have overheard. You recall, I need to put him on chemotherapy. Can the chemotherapy be done at home? No, let's wait. Let's check out his EGFR mutation status. Turns out he has EGFR mutations, then you can take him home and administer, or not only take him home, but also administer chemotherapy orally. Chemotherapy is usually injectable chemotherapy. So in this particular case, it's not even injectable, so there's no pain. You just take a pill in the morning. We're talking about EGFR. This is called epidermal growth factor receptor. I won't get into the biology of it. We now know that this is overexpressed in the cells of lung and breast cancers, and that eventually leads to uncontrolled cell proliferation, again a hallmark of cancer. This is how the protein looks like. It's beautiful. It looks beautiful. It's very well decorated in this particular domain called tyrosine kinase domain, and it's essentially there in this particular beautiful part of the protein where these alterations take place, and that causes lots of problems. Now, once you understand that, once you understood that that's where the problems are, then people started doing research, scientists started doing research, how do you block that? And so they came up with a selective so two things. One is they came up with a blocker of that particular protein. That blocker is called afatinib. It's selective for mutations in that particular domain, the beautifully decorated domain of that protein. And it's also not only selective and blocks the protein, but it's also orally bioavailable. Not all, sometimes you have to take injections to get treatment. Sometimes you can pop a pill in the mouth and get treatment. So when not every medication is bioavailable by putting it in the mouth. Sometimes you have to put it in the bloodstream. So when it's orally bioavailable, then you can pop a pill and get yourself treated. But again, bioavailability is a major issue. In this particular case, it is bioavailable. So if you look at the response rate, not only it's bioavailable orally, afatinib, but its response rate is also higher. The standard procedure used to be, before afatinib came into the market, the standard procedure used to be based on a platinum drug, which is is called cisplatin. It's injectable. It's not orally bioavailable. It's injectable, 23% response, but not when afatinib came to the market, it became orally available. So it's much easier to, compliance is much easier, less painful for the patient, and also the response rate is so much higher, more than 2 times higher. I'll skip this. The last part, last 2 minutes of my story. This is all about after cancer has set in, we are trying to understand and trying to prevent. I'm trying to treat the cancer or prevent it from progressing. What we would really like to do is to be able to stop it at a precancerous stage, even before the frank cancer has developed. Not everywhere do you have a clue about frank cancer having been developed, especially when when the organs are internal. It's very difficult to identify that cancer is appearing. You have a precancerous lesion and cancer is appearing. For oral cavity, where you can actually see, examine the oral cavity, you can actually find out that there are precancerous lesions. These precancerous lesions are called leukoplakia. The earliest precancerous lesion is called leukoplakia. And so if you, and a large fraction of leukoplakias actually will transform to malignant cancer, malignant oral cancer. So what one would like to do is that if you detect leukoplakia, you figure out whether it actually will transform to malignant tumor by doing some analysis of the leukoplakia tissue, and then if you can find some targets by which you can stop that leukoplakia tissue to transforming itself to malignant tumor, then you have really made it, right? That's what all of us would like to see happen, that at the precancerous stage we are able to stop the tumor from appearing. This is some work that we have done. We were very lucky. We did some work where we looked at leukoplakia, we looked at frank cancer and found out what kind of alterations transforms the leukoplakia tissue to a frank cancer tissue. We were lucky in 2 ways, that not only our paper was published, accepted in one of the major journals that all of us aspire to publish. It also got a cover on that particular issue. It also got an editorial in that issue. And so this is the editorial. It was written by 2 people. It was an invited commentary. The editor had invited a commentary. And what they said is that the identification of early events in a particular gene called caspase 8, which is what we have identified, allows for a potential disease intercept opportunity at the point of pre-malignant lesion identification. This whole work was done primarily by a colleague of mine, a former student, now a colleague of mine, Nidhan Biswas, who's, I introduced him to you. He was in charge of the analysis, the statistical analysis of all of the data in the International Cancer Genome Consortium project. He also was involved in this. And what we identified is that you cannot remember this. I mentioned about a list of 10 genes that are important in oral cancer. Caspase 8 was one of those genes. Now, having identified the precancerous lesions and found caspase 8 to be a major driver from a precancer to frank cancer, we now understand when this caspase 8 mutations arise. So what we did is to look at, just to cut a long story short, when you have a frank cancer, what we see is that caspase 8 is an early event. It happens in the precancerous lesion, followed by mutations in other kinds of genes that eventually transforms this precancerous lesion to becoming a frank cancer. But caspase 8 is one of the early events. So if a person comes with leukoplakia in the mouth, in the oral cavity, and we sample the leukoplakia tissue, do a DNA analysis, and we find caspase 8 mutations, we better be careful. That particular patient needs to be watched, and eventually what we would like to do is to get an inhibitor of caspase 8 and see if it works, if it stops precancerous lesions from becoming a frank cancer. So it actually will allow for a potential disease intercept opportunity at a point of pre-malignant lesion identification. So all of us dream to make cancer into a chronic disease by unleashing the power of genomics. That's what drives us. We do genomics, and it's like shooting the magic bullets, but there is nothing magical about it. The reason why we want to turn cancer into a chronic disease is very simple. If some of you haven't understood what is meant by that particular phrase, Many of the elderly people sitting here will have hypertension, high blood pressure. High blood pressure is dangerous, right? You want to keep your blood pressure in check. What do we do in the morning? We pop a pill in the mouth. We essentially take an oral tablet, and that keeps the blood pressure in check. And we are going about, this is what I do. I have hypertension. I've taken a pill in the morning. I'm pretty sure that my blood pressure well, now it may be a little high. I'm giving this talk. But usually it will remain in check. Throughout the day, we just go about doing our daily chores. So if a cancer patient, if a person has cancer, and if a person can go through exactly the same regimen, which is take a pill in the morning and go about doing your daily chores, that will transform cancer, which is a deadly disease, into a chronic disease. A chronic disease is a disease that's kept under control for a very long period of time using some kind of medication. This is what we all dream, and we use the power of genomics. There are other people who use power of other kinds of sciences in order to do this. We use the power of genomics, and I hope that through this bouquet of stories, I hope that I've been able to convince you that genomics is a good way of learning about where those driver alterations are, what kind of changes take place in cancers, and eventually that gives us a handle on finding treatment. So we we do aspire to find many more magic bullets, but there is really nothing magical about it. It's identifying a problem, hard work, dedicated work, working 7 days a week, 11 hours a day, and that's the kind of work it needs. And I'm pretty sure that many of you will actually put in that kind of labor, and we will have many more magic bullets for cancer and other diseases. Thank you very much for your attention.
My question is regarding, can we look cancer as a library, sir? In the pre-cancerous state, if we try to induce that pre-cancerous state into multiple environment, as survival of the fetus is seen in cancer, can we study about different types of more protein which we haven't discovered yet and try to use it in another field, sir? For example, aging and all.
The answer to your question is yes, we can't do that with humans. We can't expose humans deliberately to adverse environments. That's not ethical. So oftentimes what is done is, if we have a speculation that this kind of environment might actually be inducing cancer, we do that in the mouse, or we do that with cell lines. We expose the cells to a specific kind of adverse environment and see whether other kinds of proteins are start to be expressed. We can't do that with humans, but you're right that this is done, and it's done profitably as well.
Really nice to hear this beautiful lecture after several years. Actually, I heard your talk after 7 years before, actually. So NIBMG organized one. I met Arindam sir also. So sir, if a doctor or scientist say that a particular kind of cancer prognosis is very poor prognosis, what do you mean by that? For us, actually, we little know. And they say that certain kind of cancer, like maybe signet ring cell carcinoma, they have poor prognosis. And how to decide the prognosis is pure, and if genomics can help? Also, sir, how to decide if doctor, because lots of cancer patients goes, and it is difficult to do genomics for all those things. So when to decide we have to do the genomics? Because in this reason, because when my father was also undergo this cancer, there was nothing, and we couldn't go to genomics and all those things. They say that it's a poor prognosis and all those things. So they were trying several drugs, some way to work, some not. So what is the way to decide, and is there a body or something standard is there to follow such dispositives? All right,
sirs. I don't know if I use the word prognosis during my talk, but that's something that's good to know what prognosis really means. It means if a person has cancer, what's the future of this person? How long the person is going to survive, whether the cancer is going to spread through the body, and so on and so forth. So this is essentially, the future is called prognosis. And if you take a bunch of patients with the same kind of cancer, not all patients will have the same prognosis. So how do we know the prognosis? Essentially, we follow up the patients. If they have gotten treatment, they've gotten the same kind of treatment, we follow them up for a period of time to figure out what kind of problems that they are facing, how long they are surviving, and so on and so forth. And then what we try and do is we, again, try to relate their survival with the kind of mutations that they had, when they had cancer. So this gives us a handle on which particular genes may be useful, or which particular mutations or alterations may be important or involved in poor prognosis. Poor prognosis is what we are really after, because if it is a good prognosis, then the patient is going to survive for a long period of time. We are more interested in identifying what makes the prognosis poor, and can we manage that poor prognosis. In terms of, so that was the first question that you asked. So this is what we do. Didn't give you any examples, but we do have examples about on that regarding oral cancer as well. The second is that, you said that when do we order for genetic tests, and how can a patient even afford genetic tests? Well, right now, for example, vemurafenib, the example that I gave you, if the patient comes to the doctor with a particular form, with a particular clinical type, the doctor suspects that vemurafenib may or may not work, or may work, then the first thing that the doctor does is asks the patient to sequence that EGFR mutation and find out whether there are mutations in that gene. And if there are no mutations, the doctor is not going to prescribe vemurafenib. Similarly, like what we said that we identified 10 genes, and if a patient comes with oral cancer and we want to manage oral cancer based on which particular genes there are alterations, then we would ask them to get these 10 genes sequenced and then take clinical decisions based on which genes are mutated and which genes are not. So we don't necessarily need to sequence all of the genes in the genome, because that's kind of expensive. The patients also will not be able to afford that. So this kind of research actually tells you that you can actually home in to a smaller number of genes, so that the patient does not have to spend too much of money generating the genomic evidence that the doctor will use for clinical decision making. Yeah? There's a question there.
To you that you're here today. It was really an interesting seminar, and I'm also grateful to the Royal School of Bioscience for organizing this event today. So I have a small query. So like sometimes few diseases, when we go for a checkup to doctors, like for example, if one is suffering from some menstrual disorder, the doctors normally predict that you might have cancer in the future stages. So how do they predict about this? I want to know this.
So honestly, these proclamations without justification, and it is for us, and very often, at least in this country, we can't even question doctors. We can't ask, why do you say this? Because they feel offended. In the United States, that would not happen. It's within your right to ask the doctor what makes you make this statement, or say this statement. So here, unfortunately, we don't ask, but oftentimes also the doctors are making proclamations without scientific justification. So the more we do these kinds of research, the more evidence that we produce to the doctors, the better educated they will become in making these proclamations. That's what my belief is. Yeah, right now, a lot of hand-waving is done by the doctors. They will make also it's their bread and butter, right? To keep the patients in hand is their bread and butter. So again, I don't want to say very negative things, but there is a lot of negativity associated with clinical decision making in our country and in many other countries as well.
Thank you, sir.
Is that can the oral medication and injectable medication be taken together in cancer?
Can what kind of medication?
Oral chemotherapy and injectable chemotherapy.
Yeah.
Both.
And injectable chemotherapy will hasten the process of amelioration of tumors? It will not. It has its own time, so usually the doctors won't prescribe that both together. They will either, for most cancers, oral chemotherapy is not available. Mostly it will be injectable chemotherapy, but sometimes when there is oral chemotherapy available, they will prescribe that, but they will not then prescribe the injectable.
Is oral chemotherapy is available for leukemia?
For?
Leukemia?
Oh, no.
Okay. Thank you, sir.
who have a history of cancer in their family, they get really afraid that whether there is risk of having cancer in their lifetime. Nowadays, people have a little bit concept about this genome sequencing, although in a layman language, people know little bit the genome sequencing means that they are sequencing their whole, their genome. Sir, there are some genetic counseling is also going on who encourage people that by genome sequencing, you may predict the risk of the cancer. Now, as per my knowledge, a healthy individual only can give his blood, and if the genome sequencing is done through blood, the germline mutation we will get, not the somatic mutation.
Correct.
Because a healthy person cannot give the biopsy sample. It is not possible.
Cannot?
Cannot provide the biopsy is not possible for a healthy individual. Since
Biopsy of which organ?
Yeah,
There's no
exactly.
organ that's
because
affected.
since there is no cancer. So will this germline mutation of a healthy individual can predict the risk that he might have the cancer in the future?
Well, for sure, she might have the cancer if she has BRCA1 mutation.
Yes.
This is what Angelina Jolie did.
Yes.
Whether if you have leukoplakia and you have caspase 8 mutation, then the chances are high that you're going to develop frank cancer. So for some cancers, we are able to predict, and by the way, caspase 8 is not a germline mutation here.
It is a somatic one.
Is a somatic one. So essentially, only about 10% of cancers will be, germline, and therefore, to be able to do germline sequencing and predict a risk of cancer is very small. Only in big hit genes like BRCA1, you are able to predict.
Okay, sir.
Yeah?
Thank you.
There are some genes like in colorectal cancer, P53, for example. So some big hit genes will provide a lot of information, but that's small, rare.
One, BRCA2, or they become more susceptible to certain types of cancers. Do we know of any instance that when people have a certain mutation, they become immune to cancer or less likely to have cancer? Maybe because they have a certain disease or some epigenetic thing, like that makes them less likely as compared to the general population to have a certain type of cancer. Has that been observed?
So instead of being predisposed to cancer, is protected against cancer. Is that the kind of question that you're asking?
More like, does anyone, can you predict if that person will be more, like for example, if someone has a certain gene like BRCA1, they're likely to get cancer. But I'm asking, if someone has a particular gene, and we observe that they have that gene, can we say that they will be immune to a particular type of cancer?
So again, two things. Number one, every one of us has those genes. Mm-hmm. It's whether or not there is an alteration in that gene that is the issue, because alterations will lead to cancers, whether it's in an oncogene or in a tumor suppressor gene. What you're saying is that, are you using the word immune in the sense of protection, or are you using the immune in a scientific immune system, immunology perspective?
Will not be affected by the disease at all.
Okay, so you're using in a kind of English word, the word immune.
Sorry, yeah.
So what you're saying is that, can we look at the genome and predict that this person is protected against cancers? Yes. That's the kind of question that you, right now, no. Right now, no. What we have learned are only the drivers. What are the protectors, we don't know. The policemen, we still don't know. Yeah, so we can't answer those kinds of questions yet.
Thank you so much.
Thank you.
Thank you so much for enlightening us with such an informative session. So my question is, simple question is to you, sir. How this process will help in diagnostic and treatment of cancer to the human being?
What? I couldn't understand.
This genomic, the magic bullet or the genomic process of identifying the driving genes,
yes,
will help or contribute in identifying or the diagnosing and treatment of cancer.
BRAF, right? I gave you an example of BRAF, because of which the most prominent mutation, identifying that, looking at what that most prominent mutation does, we now have a monoclonal antibody or a treatment against that, right? Bemurafenib is one. I also gave you an example of EGFR mutation. So there I can give you plenty more examples, but obviously time is a constraint. So the point is that by using the power of genomics, we are able to find these driver alterations. Once we identify the driver alterations associated with some particular disease, then we try to do more research on that particular alteration to find out what impact it has on the protein that it is producing, and can we stop the negative impact on the protein by using a molecule. So this work will continue, and there will be more and more drugs, more and more treatments that will come out as a result of this kind of research.
Thank you, sir.
Thank you everyone for all your questions, and this has been indeed a great interactive sessions, and thank you, sir, for accepting our invitation and for this wonderful talk over here. And now may I request Dr. Amlan Das, sir.
Deans, senior professors. Now having Professor Majumdar giving a research talk here is a kind of red letter day for RGU. So I have been personally associated with Professor Majumdar for last 3 years before joining RGU, and I have known his dedication to science, and not only science, to popularize science and to also simplify the basic scientific knowledge to the mass. Sir, it is our sincere gratitude to you that you have take out some time from your very, very hectic schedule and come to RGU for delivering your research talk. We are very much thankful to you for your time. I want to thank our Chief Advisor, Professor M.K. Chaudhuri, and also Honorable Vice Chancellor, sir, for their constant support and vision to develop the research ecosystem here at RGU. I want to thank Professor Alok K. Budhagaye, sir, for his encouraging vision and huge dedication for science and research and also the academic environment at RGU. I want to thank Professor Anupam Chatterjee, our beloved Dean, sir, of Royal School of Biosciences. It is his vision that we have arranged the lecture of Professor Majumdar at RGU. I sincerely thank all the deans, senior professors, and the faculty members who have present here and has made this occasion very successful. And finally, my gratitude and sincere thanks to all the students of the UG and PG courses who have come here, and they have been very patient. They have listened to the lecture of Professor Majumdar. They have responded to his lecture. They have asked questions. And without your contribution, this program will not have been successful as it is. So my sincere gratitude and thanks to all the RGU family for successfully organizing and also successfully contributing to organize this event. Thank you very much.